everyone, this is our quick little video on how we see light. Let's get started.
Guys, so I'm here to talk about the anatomy of the eye. So let's get started. So first things first, you have the cornea, and that's the outermost layer of the eye. It just protects the eye. Then you have the pupil, and basically this is the dark area where you see, and all it is a hole that's letting in light, just different amounts. Then you have the lens. So the lens changes shape to bend light so that it'll focus on the fovea. And the fovea is the area with basically the best accuracy, as you would say. That's just an area of a lot of concentration of cones. We'll get to that later. Next, you have the retina. The retina is where all the photoreceptors in your eye are located, and this is all around the inner lining of the eye. And we'll get more into the anatomy. So now we're going to move on to the actual anatomy of the retina. So the retina is kind of weird because light enters through your cornea, but when it comes in, it doesn't hit the ganglion cell first. It enters and it hits the rods and cones, and those are your photoreceptors. So you have the pigment epithelium right here, and basically this functions to catch any extra light that isn't picked up by the rods and cones, so your vision isn't distorted. And this contains melanin pigment. So next you have the rods and cones. So what are those? So rods and cones are the two photoreceptors in your eyes that are most commonly known. There actually are others that have been discovered recently. So the rod is basically, it focuses on low light, like night vision, dim lighting and stuff like that. And then also it focuses on like black and white. So it doesn't really do too well with color. And also these outnumber your cones by about 20 to 1. So you have a lot more than, cones. And then you have cones. So cones are specializing in like the sharpest vision. They see color, they see all the sharp images. So they're very important. So your rods and cones basically communicate with bipolar cells that communicate with ganglion cells. And these ganglion cells eventually lead up to your optic nerve. So the light comes in, hits your photoreceptors, your rods and cones, and then those transduce signals to your bipolar cells. The bipolar cells act as a bridge, and so basically they take information from your rods and cones, send them to your ganglion cells. Your ganglion cells eventually send them up to the optic nerve, which eventually go to the brain. All right, so we just talked about rods and cones, so I'm going to show you the structure of a rod. So a rod is directly bordered by the pigment epithelium. So rods are the neurotransmitters, and they contain pigments called rhodopsin. Cones also contain various pigments, but they're very similar to rhodopsin. There's 3 different types. So within these different discs, you have rhodopsin. And so, like I said, rods work in low light. So during low light, rhodopsin is inactive, so it's binded. And rhodopsin is actually made of 2 different things. So rhodopsin is made of 2 different chemicals, basically. So you have the first thing called retinol, and retinol is a vitamin A derivative, which is binded to opsin. So in low light, your rhodopsin is inactive, and that means that you have CMG levels that are very high. What this means is you have a high influx of calcium and sodium. However, you have a low efflux of potassium, making the cell basically very depolarized. So instead of being the normal, 70 millivolts of a cell, it's about -40. However, when the rod gets, light basically, then the rhodopsin becomes active. These two unbind, so you have the retinol leaving, and it goes and enters the pigment epithelium. When this happens, the cGMP levels are low. And so now you have the calcium and sodium channels being closed off, and you still have a high efflux of calcium. I'm sorry, not calcium, potassium. This makes the cell hyperpolarized, leading to the neurotransmitter, also known as glutamate, being released and entering the bipolar cell.
So signaling processing is going to begin when glutamate is released from the photoreceptors, and it's going to go to the bipolar cells. And there are 2 different types of bipolar cells. And the light on bipolar cells are activated when the glutamate isn't being released. The light off bipolar cells are excited when the glutamate is released, and they are not being activated when it's not being released. When the glutamate is released and it binds to the light on bipolar cells, and it's going to cause hyperpolarization. and when it's not binding, it's gonna cause depolarization. And after this, the action potential is gonna be sent to the optic nerve. Okay, so the action potential is going to travel down the optic nerve where it's gonna go to the central nervous system, and nerve fibers from each eye are going to cross over to opposite sides of the brain so that the two images are combined so that you can form one 3D image. And then the axons are going to leave the optic chiasm, and they're going to go to the lateral geniculate body of the thalamus, which is organized into 6 layers that all correspond to different parts of the visual field. And these processes are how you see light.